Hello and welcome back to Survey of Biology as we continue our study into Unit 11 on genetic diversity and variability in populations. And so if you remember, we looked in the first section, this is section 3, but in the first section we mentioned what mutations were and that they're just a change in the normal base pair sequence. So any errors, any mistakes in the normal sequence of A's, T's, C's, and G's is a mutation. And as we're going to see, some of them can be very minor, but some of them can be very major indeed. So here's just an example. If you look in the middle, there's Yoda. And that's a cat that had a mutation and was born with 4 ears. And that one's pretty easy to see. And if you look on the sides over on the left-hand side, in the upper left, you'll see there's a duck that is missing the webbing on his feet. That's a mutation. And over on the lower right, you'll see there's a barn owl. This one is white and one is gray. So those are mutations in the genes that allowed for different alleles that coded for different colors. So let's look at what mutations are and how they can happen. So mutations can be a single base pair, what they call a point mutation, or they can be larger scale changes like what we'll see as like a frame shift. And these, remember, mutations are random, and they accumulate over time in populations and individuals can be passed on. And the big thing to remember about mutations is they're changes in the DNA but they affect the expression of proteins. So if we go back for a second, look at Yoda there. So how did a change in the DNA give Yoda 4 ears? Well, it was a change in the protein, a growth protein or something like that, a hormone that did that. So if you remember, so mutations are changes in the DNA, but they're going to affect our expression of proteins and express how we look, the phenotypes. So here's what mutations actually are. So if you start over here on the left, here's the DNA before replication. And if you remember, it's a complementary to double-stranded. So A's go with T's and C's go with G's. And when we want to replicate it, the DNA will split. And we have the top one here. And this one's correctly replicated. The complementary base pairs all fill in, and we get an exact duplicate of our original DNA. But if we look on the bottom, we've got an error at this location right here. So this is what we would call a point mutation. There's one difference. And here, a T accidentally gets put in where there should have been a C, and we have a mismatched pair. Now, in many cases, the good news is this gets caught. We have proofreading enzymes that take care of this, and we fix it before anything ever comes of it. But sometimes it's missed, and we go through and we end up with what's called a mismatched pair. And this is how mutations can occur. So now there's different types of mutations, and I apologize, this slide might be a little hard to read. But if we look at the normal gene up top, what happens is we start reading, and there'll be what's called a start codon. So that's where we start reading, and then there'll be an end that's kind of like the period of DNA. It tells us where to stop. So if we read this, it reads, the man saw the dog hit the can. And that is what that gene would express. Now what happens is in a point mutation, as we saw before, there's a replacement of one letter for another. So now if we have a point mutation, we can read this, but it says the man saw the dot hit the can. We can still read it, but it has a very different meaning. So this is something like sickle cell anemia. Anemia is an example of a disease that's one base pair difference. Now we can have what's called a deletion. Okay, so here's an example where we have deleted a whole section. So the man saw the hit the can. Now we can kind of read it, but it's missing something. Okay, so that's called a deletion. Then we can also have what's called an insertion. And here's where we insert a whole new thing. So now the sentence reads, or the gene reads, the man saw the fat dog hit the can. We can still read it, but it has a different meaning than the original gene that was being expressed up here. And all those are relatively minor for the most part, not all of them. But what's really bad is if we get something called a frame shift. And a frame shift is when one base pair from the original one gets deleted. So here we had deletion in the middle, but here we had 3 letters. So we had a whole word. What if we have only one letter? In this case, the D. Well, the way DNA works is that it's read in groups of 3 to make the proteins. So now the man saw the og, it, heck, ain, nu. The rest of the sentence is nonsense. It doesn't make anything, or actually it's called missense. So starting right here, everything downstream can't be read, and our protein is very, very different. This is the worst kind of mutation because everything downstream from there is affected by that. So here is a picture that shows this in a little bit more detail. So if we look in the upper left hand here, here's our normal peptide: phenylalanine, tyrosine, alanine, arginine. Those are the amino acids that this DNA is coding for. That's going to make our protein. Sorry about that. There, let's get rid of that. Okay. Now, there's something called a silent mutation. And in a silent mutation, if we look, we're supposed to have AAA, but we've got AAG. And in some cases, like this one, it still codes for phenylalanine. So the protein that we produce, the polypeptide, there's no change, and that's what's called a silent mutation. And there's a good chance that we all probably have one of those in our genome right now. Now, what happens if we have a change right here where we're supposed to have alanine, but we get glycine instead? So you see we're supposed to have a G, but we get a C instead. It codes for a different amino acid. We still get a protein, but it's a slightly different sequence. That's called a missense mutation, kind of like a mistake. We get something, we get an answer, but it's not the right answer. Okay? And sometimes that can be bad and sometimes not so bad. And then here's another example up here. So where we have, right here, we're supposed to have an A, and we get a T. And that codes not for an amino acid, but for a stop codon. And that's bad because now the polypeptide, the protein synthesis ceases. This is called a nonsense because we don't get a protein. There's no protein produced. Okay. Now, as we go through, here's the frame shift that I talked about. And frame shift can happen in 2 ways. One is, what if we insert an extra base pair, or what if we delete a base pair? In both cases, everything, the reading frame gets shifted down, and we end up with very different proteins, usually major differences. That's why frame shift mutations are so bad. So there's an idea of what mutations are and some different types of mutation. Now, what can cause them? Well, mutagens cause mutations. So a mutagen is a physical or chemical agent that changes genetic material. It causes mutation. And the big 3 that we're going to talk about are chemical substances. Tobacco is probably the #1 carcinogen out of all substances in the world. Radiation in all its different forms. There's nuclear radiation, there's ultraviolet radiation, there is X-rays, and then also viruses can cause mutations that can cause cancer. So HPV, some of you may have been vaccinated against that, is a virus that can cause cervical cancer in women. And so there is an example of how viruses can cause damage to your genes and cause a mutation. So radiation is the big one that we'll talk about. So here's an example. You've all probably seen a Godzilla movie in some form. They've been around for years. But radiation can take the form of nuclear waste. It can be ultraviolet light. That's why we should wear sunglasses and put sunscreen on when we go outside during the summer. Or X-rays. That's why they put the big lead vest over you when you get X-rays at the doctor's office. And one thing just in the last couple of years since the Fukushima meltdown in Japan is that they're finding butterflies with genetic damage around those nuclear power plants. So about 12% of the larvae have abnormalities because of exposure to nuclear waste from the Fukushima generating plant over there. And so this is something that will continue to be studied as we go through. So some of the mutations that they're seeing in these butterflies are smaller wings and damaged eyes. We talked about ultraviolet radiation. And so UV light is ionizing radiation because it has the ability to create ions, literally knock electrons out. So it can come in and it can strike your DNA, and it can cause mutations in that DNA that can lead to melanoma, skin cancer, and other things. So there's also X-rays that we talked about before, that you don't want to get too many X-rays, because the same thing, it's an ionizing radiation that can come along, and if we look here, it can cause breaks in the sugar phosphate backbone of DNA and cause damage that can lead to cancers. So that's the big thing about these mutagens, is that many of them are carcinogens because they can also lead to cancer. So there we go in our quick section about mutations and what can cause them. So thank you for your time, and we'll see you soon for our last section in Unit 11.